Good evening. It is your professor, Dr. Wood, again, talking about a couple types of research. Again, some of these videos should be just review for you in terms of stuff we learned back in Epi Bio. But as some of you might not have retained 100% of that information, shame on you. But I'll extend a little bit of grace here and cover some of the things I think are really pertinent. The reason why I'm covering this stuff again is because I believe it's important to have some familiarity with, especially like types of studies, some basic understanding of the stats, because when you're going to start pouring through your different research articles, you need to be able to kind of understand what they're doing, what types of studies they're doing in terms of like study design, and kind of what you're able to take away from that based off of what they're doing essentially. So let's look at kind of this diagram here of kind of all research in its various forms here. So basically you start off, there's all truth in the universe. I guess if there is any truth there to be had. But again, we're going to find that typically we start off with a problem, right? So say, for instance, with our research papers we're going to be writing, we're curious about a question, like what's the best way to manage this? Are there any, co-inheritance of different disease states together? I mean, you guys have really come up with some good ones so far. But you're defining the problem essentially, right? You're trying to determine what sort of clinical issue there is and how you want to go about solving it essentially, right? And so anyway, so at this point is where you're going to be developing your research question, along with that your hypothesis and also your null hypothesis, right? We'll talk about those a little bit later on in a different video. But first off, what you're going to find is that you start off with a literature review. So similar to what you're going to be doing for this paper specifically, you may find the answer is just already out there, right? So once your solution is discovered, you're kind of done. So if I have a question about how I should manage a thrush in a patient, I can just go and look up an up-to-date article. It'll say, here's how you treat thrush. Do nystatin, do whatever, fluconazole. And the answer is already done, right? Now, let's say, though, that we don't have the answer in the literature. What you're mainly going to find is that for the most of your research questions, there's not a definitive answer that's already out there. And that's a good thing, because if there was a definitive answer, you wouldn't have a very good paper. You'd have to come up with something else. So at that point, there's a couple of different things you can do, right? So you can develop and design a methodology to try to show what you're looking for or try to answer the question that you've posed there. Based off that methodology, you do your data collection. You'd have information you'd be collecting there. You would then analyze that data. This is where your biostatistics come into play. And then you have your results, right? At that point, you're going to start to interpret things. You're going to draw some conclusions, and then you disseminate it out, right? So whether it's going to be a publication to a journal, whether it's going to be a poster that you present at a national meeting, could be lots of different formulations. But there's no point in keeping research to yourself. You're supposed to disseminate it out to the world so people can look at it, evaluate it, replicate it potentially, or act on it. So again, at that point, they'd be reviewing your literature and trying to find out the solution to their problem. So that's the general flow of things. Well, how do we design studies in order to get this information, right? How do we get that information to where we can draw some sort of meaningful conclusions from it? You're going to find that depending on the type of study that you may or may not be able to take a whole lot of information from that, especially when you're in terms of causation, right? So again, is this causing this? With certain study designs, you really can't tell that, or at least you can't draw that strong of a conclusion. So getting into it, we can start off at the base of our pyramid here with sort of research we're not going to be dealing with too much. A few of you, depending on your study, your research topic, you may look into some animal research here. You may look into in vitro test tube research. Some of you have probably done some of this test tube research before. So if you've ever worked in a lab, you've been culturing cells, any of that sort of stuff, that's what we consider to be kind of the bench research, right? From that, though, this is where you can develop some things like ideas and opinions and editorials and whatnot. Really, where we're going to be stepping in terms of doing our literature review are going to be things like case reports, right? And you guys can write case reports, and it's actually highly encouraged. If you're out there on rotations and you see a really interesting patient, if you see something really kind of neat, you can develop a case report, write it with your preceptor, or write it with whoever, and then get that published. I did that during my fellowship. I found this really interesting case of a patient who absorbed vancomycin enterally, which was kind of odd and novel. We sent that to a pharmacotherapeutics journal, and they accepted it, and they got published, right, just off a single case report. They're going to look at some downsides to case reports and how they're not really that great for getting a lot of conclusions from it, but there are still some utility for using case reports, right, for publishing these things. From there, you're going to find our case series. Then we'll have things like case control studies, cohort studies, and finally you're going to get into your randomized controlled trials, right? These are going to be the mama jamas of getting good causation sort of information, right? We'll talk more about that in a second. And then finally, this is where you get into your systematic reviews and meta-analyses, right? So they're going to take all this different information, all these different cohort studies and RCTs, and kind of combine them all together to hopefully come up with a nice unified theory. If in your course of research, if you look up the Cochrane database or the Cochrane Review, that is what that is. Essentially, they just do meta-analyses for different sorts of clinical questions. That's another resource I'd highly recommend you guys check out, depending on what your topic is. They may have done a Cochrane Review of something similar that may be able to help guide you a little bit in your study or in your research. So getting into the in vitro research, as I mentioned, this is test tube research. It's good for theoretical results, which may be able to apply back to humans, but unfortunately, things done in a test tube may not directly be applicable to patients, right? Patients are much different when you're in vivo than you would see with, say, culturing human cells to test in vitro, right? But you can get some useful information. But you probably won't have too many of these studies come up for your topics, but it depends on what your topic is to see if this even comes up. From there, you can have animal research. This is usually done in mice and rats, but there have been other types as well. You can see like monkeys, rabbits, all different types of animals. Fish sometimes can be used. But again, some of these results can be applicable to humans, but not always one-to-one. So just because you see, say, a drug is really effective for treating cancer in a mouse does not mean that that necessarily applies one-to-one to humans, right? And so it can serve as a proof of concept. It can be something that will help to sort of lay the groundwork for doing future research in humans. And very frequently, especially in drug trials, they will do, drug companies will do studies in animals first to see how they work, and then they move on to humans once they show that, hey, there's safety and efficacy here. Okay, and then once you have that human data, you should be able to kind of apply that back and see if how synonymous it is, right? To see how much it see what the differences and the similarities are between the two there. So what we're going to be getting into is going to be initially case studies, right? So usually this is going to be an N of one, or basically you just have one patient that you're looking at, right? It's a single case. Again, this is usually when something novel or rare has occurred. And again, the way you find out if something you're looking at is novel or rare is to look in the literature, right? So if there really hasn't been very many publications regarding the topic you're looking at, then you're good to go, right? That's something that is new, and you can get that published potentially. It's unlikely to be applied to other patients because it may be a very unique sort of scenario or maybe a very rare sort of scenario. But what's interesting is that it may actually prompt further research. If you see something kind of interesting, well, maybe that's happening to other patients too, and we just didn't have our radars up for it, okay? Usually what you're going to find in case studies is that you'll also write a review of the topic as well, or you may report on similar cases that have occurred here. So just as an example, you can go to an article, if this will work. So I'm going to log in here. And right, so this is that article I was mentioning that I had written back when I was in fellowship. And so this is actually, I was on a picky rotation up in Jacksonville, and these were two of the preceptors I was working with. And basically what happened is that, we found this patient that was really kind of interesting and unique. And so we decided to write it up, right? And so one of the things you'll find is we include an introduction sort of talking about, why we think this was really kind of interesting and why this is sort of a unique, novel thing. We go through the case presentation, right? So we kind of talk about what happened for the patient, talking about the course of events here. And then we have some tables here talking about vancomycin levels, right? I don't want to bore you guys too much with this. But then finally, there's a discussion here. So we kind of go over previous cases where this phenomenon was reported. And we talk about how this is kind of a novel thing happening for our particular patient who was on a lot of immunosuppressants. She had very severe C. difficile colitis, all these sorts of things. And then we conclude it out. So again, it was a novel thing. It's pretty easy to get published overall, and I definitely recommend if you guys see something really interesting on rotations, talk to your preceptor and be like, hey, is this unique, novel? Do you think it'd be good, for a publication? And they'll be like, yeah, sure, go ahead, write it up. If they get their name on it, that's a bonus for them. You do all the work, but of course that's academia for you, right? So anyway, getting back to it, moving on from a case study, we have a case series. This is very similar to case reports, but usually it'll include 2 or more cases. Generally, you may find, say, between 2 and, say, a couple dozen. Some cases, it just depends. This is better because in terms of showing some sort of correlation with things, because it shows a trend, right? A single patient is just a data point, but when you have multiples, that's where you get trends or commonalities between them. And you may start to think, well, maybe this is applicable to other patients, right? Maybe I can apply it to patients I may be seeing on rounds or wherever I happen to be. Note here as well is that the difference between descriptive and inferential statistics. Remember, descriptive statistics are just going to be things that kind of describe who you're looking at or who your patients are. This is going to be more seen with these case series where you can talk about means and averages and modes and things like that. But you can't really get into the inferential statistics. That's where you're going to start to be doing things like ANOVAs, t-tests, etc., in order to actually draw conclusions from that. You can't really do that with case series because you haven't really done any actual formal controlling for the scenario. You haven't really done any formal matching, anything like that. And so you can really describe the cases, but you can't really draw a whole lot of conclusions from them. If you look at that Coral Snake article that I'd published, you'll see we talk about it as being a retrospective case series. So we drew a ton of different cases. In that one, we were actually looking at, I think I had over 500 cases or something like that, if memory serves. And by looking through all of those, we're able to draw a lot of information in terms of what the averages were. But it was hard to do formal comparisons between groups because, again, the study was never designed to do that. Okay, and again, just because you see a correlation does not mean that that is causation, right? We'll talk about some other study types that can do that. Now, case control studies, generally these are going to be retrospective. Remember, this is where you're going to be matching patients based on the outcome. So, for instance, if I had patients who I had lung cancer, and I wanted to see which sort of risk factors put them at risk for lung cancer, I would take those patients with lung cancer, match them with similar patients without lung cancer, without the outcome. So they would be similar ages, they would be similar genders, similar backgrounds, but they would say not have lung cancer. And then you look backwards in time to see what risk factors were more prevalent in the lung cancer patients versus those that weren't. And lo and behold, you probably would find something like more of them were smoking, right? And so that would be a risk factor. And that's where you're going to get things like odds ratios and whatnot. I'll cover the stats review in just a little bit. But remember, the outcome is already known for these patients. You're matching them based off their similarities to your controls that did not have the outcome, and then you look backwards for those risk factors, okay? Again, this is good for associating those risk factors with a particular outcome, and we're going to be using odds ratios. If you're using something more prospective like a cohort study, that's where you're going to be getting into things like risk ratios, right? So minor point, but it's something you'll see there pretty frequently if you look at a case control study. Compared to that, we have cohort studies where patients actually get broken up into their risk factors. So in this case, you could do a study where you took patients who are smokers, match them to controls who are non-smokers, and then follow them for an outcome. In this case, it might be lung cancer, and you would probably see the smoking group had a higher rate of lung cancer than the patients who were non-smokers. Okay, this gives you more ability to draw cause and effect. It's not the best ability, but you're starting to see more. You can draw stronger conclusions, right, and be more confident when you apply that to patients outside of your study group, right? If you were to apply that to real life patients. Not the same as a randomized controlled trial because patients don't get randomized to one group or another. They get randomized based off, or they get placed into groups based off of their presence or absence of a risk factor, okay? Again, these can be retro or prospective. Most of the time they tend to be prospective. And then finally, we have the more complicated, the much harder to run, the more expensive, time-consuming, randomized controlled trials, right? This is where groups truly get randomized to either control or experimental groups, and then you follow them for an outcome based off whatever intervention you're going to have here. So in this case, you could have a study where you take people, say all of them are non-smokers, you're going to be randomly assigning them to group A, group B, people don't smoke. A people, you make them smoke, say a pack of cigarettes a day, and then you follow them for lung cancer. That would be a very good way to draw cause and effect to say like, well, look, we have these patients who never smoked before. We gave them all, cigarettes a pack a day for a year. We followed them to see who got lung cancer. That would not be very ethical, as we'll talk about in another video, but you're going to find that would be a very strong case to say, well, yes, this is the one thing that we introduced. This was the one variable, and look at all these people who got lung cancer now. Obviously, that's not going to be a very good study there, but you can see the idea. Blinding is really important for this as well. So, for instance, when you hear about a double-blinded randomized controlled trial, that generally means that you're going to find that the providers, the people who are actually diagnosing and getting the people into the study, are blinded, meaning they don't know what the patients are getting. The reason why they do that is because they want to try to limit bias. Because if you know a patient is getting active drug, say versus placebo, you might be more likely to have favorable outcomes for, say, the experimental group, the ones getting the active drug. The double blind is where the patients are going to be blinded from the results. Okay, the patients, when you get them into the study, you have to tell them, hey, listen, you might get active drug or you might get placebo, right? The patients don't know, and they might not know until after the study is actually completed there. So that's a double-blinded sort of methodology there, okay? Now, again, imagine if you had, say, a medication that was being used here. Usually the one person who is not blinded is generally going to be like your research pharmacist. So, for instance, we were doing a study at Nemours for patients with spinal muscular atrophy, or SMA, and it was a very, very intensive sort of blinding procedure because we had it to where patients were either getting medication that was being applied intrathecally, right? So being applied directly to the spinal cord through a lumbar puncture. They were either getting placebo or they were getting active drug. And so it was very, very important that we maintain the blinding there to make sure that patients had no idea. The parents had no idea. Of course, if you're a parent of a child with a debilitating disease, potentially life-threatening, you want to know if they're getting active drug or not, right? And so they would do all kinds of things to try to find out, but we had to remain very steadfast. You had to keep your poker face on to make sure that they never saw whether you knew if it was the actual drug or not, right? But anyway, so that goes into the blinding, but usually this has very strong evidence of cause and effect. And so this is kind of the gold standard in terms of showing causation for research. And then finally, you can have a systematic review, a meta-analysis. Now, systematic review is more just kind of going through and just stating, here are the different pieces of literature, here's what they say, here's what kind of conclusions we can draw from it. Meta-analysis is going to be a little bit more intensive. That's where those Cochrane reviews I mentioned are, where they can actually go through and they'll pull these different studies together. And depending on the homogeneity or the heterogeneity of them, right? Homogeneity just meaning how similar they are together. Heterogeneity meaning how different they are. So, for instance, it's really hard to compare two trials together if one was in adult patients and one was in child patients, right? It can be very difficult to do that. So they take that into account when coming up with their conclusions there. And generally, Cochrane reviews are very, very good for looking at specific topics. Again, they may find that, hey, there's just not enough literature here to really draw a strong conclusion one way or the other, but it is what it is. So those are the main types of studies you're going to be running into. Again, randomized controlled trials are going to be difficult to run. They're very expensive and time consuming. That's why you end up seeing a lot of things like case control studies and cohort studies, things like that, where the data is a little bit easier to collect. Anyway, so there's lots of study types, and it's important to know kind of what you can take away from it based off the different types of study groups, based on whether it's retrospective, whether it's prospective. Just know that RCTs are going to have the strongest level of evidence to show true causation. You can take much more stock in what they're saying when assuming it's run correctly, but you can take much more confidence in their answers there. And then case studies tend to have the weakest level of evidence. Just because I have an N of 1 saying, like, look at this, what happened. That patient could be 1 in a million, could be 1 in a billion type of person. It's impossible to say it would ever happen again. Versus if you have a randomized controlled trial, we have thousands of patients, you can feel very comfortable with that as a strong result there, and you can take some stock in that, right? Anyway, so those are the different study types you're going to run into during your research. If you have any questions, let me know, and I'll talk to you next time. Thanks a lot.